Hey everybody, this is the cell lecture. This is chapter 3 in the Marieb and Hone Human Anatomy and Physiology book. We're going to try to do a quick run through of this chapter in about an hour. So it's not going to be the most in-depth cell lecture that you've probably ever had, especially if you've taken a biology class or something like that. We're going to try to cram that whole biology class into about an hour, so we're obviously going to skim through some stuff. Let's dive right in. So the cell is the smallest living unit. There are, of course, smaller units and smaller building blocks in our body, but they are living. They aren't capable of dividing. They are able to produce their own proteins and regulate their own metabolism. So the cell is the smallest living unit. And that brings me to cell theory. There are 4 concepts collectively known as cell theory. The first is that a cell is the basic structural and functional unit of living organisms. The second is that the activity of an organism depends on both the individual and the collective activities of its cells. The third is that the biochemical activities of cells are dictated by their shapes and forms. And the fourth is the continuity of life from one generation to another has a cellular basis. The first cell was observed in the 1600s by 2 German scientists. If you can imagine discovering cells in the 1600s, that was probably pretty revolutionary. And then since the 1800s, that's when we really started to learn a lot about cells, and that's when cell theory was developed. For our purposes, I won't be asking you about that on a quiz, so let's get into some more exciting stuff, which are the 3 regions of the cell. And this photo here is just an artist's interpretation of a cell. There isn't necessarily a cell that looks just like this. This is just sort of the generalized cell. It shows all the parts that most cells have. So let's take a look at the 3 regions of this generalized cell. The first is the plasma membrane, then we have the cytoplasm and the nucleus. So I think you guys probably all know what the plasma membrane is, or the cell membrane, and then the nucleus inside the cell, and everything in between those 2 structures is called the cytoplasm. So that includes the cytosol, which is the fluid inside the cell, and the little metabolic machines floating around in it called organelles. Let's go ahead and look at the plasma membrane. There's actually a bit going on in the plasma membrane. It seems like this simple little structure. There's a lot to it, and we're going to talk about that. It's actually, if it wasn't for the properties of the plasma membrane, cells could not exist and could not function. So the plasma membrane displays or is described as having a fluid mosaic model. The idea there is that the membrane is fluid, meaning it's flexible and things can move around within it. And it has a mosaic of different compounds and structures. There's peripheral proteins and phospholipids and cholesterol and glycolipids and glycoproteins and antigens, which are glycoproteins. Anyway, so lots of different things floating around in this fluid mosaic. This slide is specifically talking about the lipids of the plasma membrane. So the most common in terms of number and mass would be the phospholipids. So in this picture, the phospholipids are these little pink spheres with 2 green tails. Now, phospholipids are really what defines the way the plasma membrane works. So you have this, let's get a crayon here. You have this phosphate head, and I'm a terrible drawer, I apologize. And you have these 2 lipid tails. This looks something sort of like this. The phosphate head has a charge gradient on it. So what that means is one side is slightly positive, and one side is slightly negative. There is a charge gradient. So that means it's going to be attracted to water, because water is also polar. So the negative region of the water molecules will be attracted to this positive region of the phosphate head, and vice versa on this negative head. So that means that this side of the phospholipid is hydrophilic. It likes water. The lipid tails have no polarity. They're nonpolar. That means they like lipids and they do not like water, so they'll be repelled by water. So if you've ever seen, maybe you put I don't know why you would, but oh, here you go. You put olive oil and vinegar together and they separate because the vinegar is polar and the olive oil's nonpolar. It's the same idea here. So the oil and water doesn't mix basically. So why is this important? Well, if you were trying to come up with a way to arrange a bunch of phospholipids, this is going to be tough to draw. So say I'm going to write H2O. Yes, that's an H there and H2O here. Okay, so this over here is representing the extracellular fluid and this is the intracellular fluid. So if I was going to draw a cell, how could I draw it to make the phospholipids happy? This is always so much easier when I'm using a paper and pencil in front of class. So let's say we draw a ring, which makes sense, cells are spheres, right? So I'm going to draw a ring of phospholipids that is encircling this intracellular fluid. Okay, and here are the phospholipid tails pointing out away from the intracellular fluid. Does this satisfy the requirements of the phospholipids, of what they like? So the phosphate heads want to be immersed in water. They want to be facing H2O, and they are, right? There's intracellular water inside. So they're happy. I'm not going to draw all of them. So they're happy. The phosphate heads are touching the water. The lipid tails need to be touching other lipids. They do not want to be exposed to water, and guess what? They are, because the extracellular fluid is mostly water, so they're not happy. So this is not the way the cell membrane would work. Can't work that way. So let's try something different. What if the lipid tails are facing in? Well, you can probably already start to see we're going to have a similar problem. So the phosphate heads are happy. They are exposed to water, right? They're exposed to the water in the extracellular fluid. The lipid tails are not happy because they are exposed to water and they don't want to be. So there's really only, so that's not going to work either. There's really only one way that phospholipids can arrange. And it's not like some magical force does this. It's their own internal properties of polarity and nonpolarity that make them automatically self-assemble. So if you were to drop 1 billion phospholipids into a cup of water, they would automatically form cells. Now, I don't think anyone's ever tried that necessarily or tested it. Maybe they have, I'm not sure. So I'm not sure if that would form one big cell or many little cells or what, but their electrical and this is drawn all askew, sorry, but their own properties would force them to arrange into a bilayer. Now, bilayer just means there's 2 layers of these phospholipids. The phosphate heads are pointing in towards the cytosol and out towards the extracellular fluid. And the lipid tails are facing each other. And guess what? Everybody's happy. The lipid tails are not exposed to water, and the phosphate heads are, both layers. And I'm not going to keep doing this because it's taken too long, but you get the idea. It forms and this picture is much better down here, it forms a lipid bilayer. Now what this means is that things that are polar, like large polar molecules like glucose, for example, they can't pass through this layer. They can't pass through this layer because they are polar. And the core of the membrane is nonpolar, so it repels polar molecules. Now, lipid structures, simple lipids, are able to pass through. They're able to float right through this lipid core. So that means that this membrane, because of the phospholipids, is semi-permeable. It's permeable to some things more than others. It's completely impermeable to some things. So that's all due to the phospholipid. The structure of the membrane is due to the properties of the phospholipid, and the semi-permeable nature is due to the phospholipids. If this cell were to tear open for some reason, it would just reassemble. Again, because those phospholipids need to be arranged in this way. Their electrical properties require them to be arranged in this way. The next structure is glycolipids. It looks like they're mostly showing glycoproteins. I see proteins with little sugar groups on them, but there are glycolipids as well. These are lipids with sugar groups, and these serve as cell-to-cell identifiers. We have cholesterol. Cholesterol's in yellow. Cholesterol makes the membrane more rigid. Then we have lipid rafts. About 20% of the membrane surface has lipid rafts. So these are saturated phospholipids, so they pack together a little bit more tightly than the regular phospholipids. And we're not sure exactly what they do. I mean, we are. We know they're platforms for concentrating certain receptor molecules for cell signaling. But beyond that, you'd have to start digging pretty deep to find out specifically what they do and the different type of lipid rafts. So we're not going to go too deep into that. Second slide here in the plasma membrane talks about membrane proteins. So the last slide was lipids, this is proteins. We have a few different kinds of proteins. Any protein that is within the lipid core of the cell membrane is going to be called an integral protein. Those can span the entire membrane, in which case they're called a transmembrane protein, like this one right here, where there's a part sticking into the extracellular fluid and the intracellular fluid. Then we have peripheral proteins. So these are proteins that are on the periphery, either inside or outside. Those are called peripheral proteins. And then we have this collective structure called the glycocalyx, which is all of the sugar glycoprotein, all the glycoproteins of a cell. So here's a picture of, you can see a purple protein with a green sugar chain coming off of it. So this serves sort of as a cell's fingerprint. It also makes the cell a bit sticky actually, so it can play a role structurally because all the sugar is sort of adhesive with other cells. But primarily it is like a cell's fingerprint. These structures function to help other cells recognize one another, or recognize a cell as being foreign, or recognize the type of cell, that sort of idea. It's cellular identification. This next slide, we're going to talk about cell junctions. This actually becomes so important in other lectures. When we talk about skeletal muscle versus cardiac muscle, this is important. When we talk about which layers of the epidermis are more structurally sound or tougher, we're going to talk about desmosomes. When we talk about the lining of your digestive tract, we talk about tight junctions. So this is an important concept that we will circle back to in other chapters. So there are three basic categories of cell junctions, and they're here on the screen. The first is tight junctions, and that is actually referring to this picture right here. So if you look at it, it really looks to me like a stitch, and that's basically what it is. So if you picture two cells next to each other, or let's do this, picture a sheet of 1,000 cells. Now there's gaps in between those cell membranes. So if you were to pour water onto that sheet of cells, the water would just leak in between them and leak through. But let's say you wanted a sheet of cells to not be leaky. Let's say you wanted it to be an impermeable barrier. You would stitch the cell membranes together. And that's what tight junctions are. They're basically stitching adjacent cell membranes together so that that sheet of cells becomes impermeable, or less permeable at least. So an example of where you would see these would be the lining of your digestive tract, for example. And you might think, well, wouldn't we want the lining of our digestive tract to be leaky? And no, we wouldn't, because we would not want those acids within our stomach or intestine to leak through to the connective tissue below and degrade it. We don't want that. We want the sheet of cells to be impermeable, and then we want to be able to select what we want to absorb. So, okay, we want to absorb the sugar, the proteins, the fats, and that's the job of the cells, of the microvilli of those columnar cells inside your digestive tract. We don't want it to be leaky. So Anyway, so that's an example of where we might see tight junctions. The next one is desmosomes. Desmosomes are pretty cool. I use the example of a rivet all the time, but I found that people don't really know what a rivet is. So if you know what a rivet is, I'm proud of you. Number one. I've been meaning to do this forever, and I just thought of it right now as I'm recording. So I'm going to pull up a picture of a rivet. Okay, so this is a rivet before it's actually used, before it's actually a rivet gun is applied to it. So that's it pre-used. So this here is a rivet about to be used. Let me show you. Control V. And you're still probably not understanding this, but so here's two pieces of metal. Let's say we're building an airplane, and there's two pieces of metal. We drill a hole and we put this rivet in it, and then we slide this rivet gun over it. And what's going to happen is this gun is going to pull really hard on this shaft, and it's going to flatten this side of the rivet until it looks like, and here's all three steps, until it looks like this. So it's sandwiching the two sheets of metal together. And if you've ever seen like an old timey airplane, you've definitely seen rivets, or even actually new airplanes I've seen rivets on. So they look like, ignore the text, they look like these little bumps. And those are rivets forcibly holding two sheets of metal together. And that's really what desmosomes are. Except desmosomes are even better. Here they are in this picture right here. So you can see two cell membranes, and then you see these two protein discs sandwiching the membranes together. What makes desmosomes so much better is that if you see these little protein filaments, what this picture isn't showing you is that those filaments are integrated with the whole cell skeleton. So it's not just the cell membrane being sandwiched together, it's the actual skeletons within each cell that is being riveted together. So desmosomes make tissues much stronger, epithelial tissue specifically, but they make tissues much stronger. Gap junctions are basically tunnels between two cells. So they allow electrical and chemical messages to move from one cell to another. And here's a photo right here. You're going to see that in heart muscle, for example, where you want the electrical current to move from cell to cell. You'll see tight junctions in the digestive tract. I think we mentioned that. And you'll see desmosomes in a lot of different places, but one spot would be your epidermis of your skin. You don't want your skin to be pulled apart easily, so there's a lot of desmosomes. All right, we're getting into transport processes. This is where I'm going to start to skim. I'm not implying that it isn't important, but we could get stuck in the weeds here. So I'm just going to talk about the difference between osmosis and diffusion, how they work, and keep it pretty basic. So let's actually, osmosis is first, but let's look at diffusion. So diffusion is just this idea that let me try to give you an illustration. So if we were to drop a sugar cube into a bucket of water, we would expect it to dissolve, right? So dissolve is one word for what's happening. Diffusion is another word. So so let's look at this picture here. For diffusion. So here we have this sort of U-shaped beaker, which is kind of cool, and we have, let's say, like a cell membrane dividing the left and right half. So assuming that these solutes, these red dots, that's solute, in this case it's sugar, assuming it can pass freely through that membrane, we would expect that those, it's more highly concentrated on the right, so we would expect that those molecules would diffuse to the left. Until eventually there's the same concentration of sugar in the water on the left and the right. That's diffusion. Just like if we were to drop a sugar cube into a beaker of water, the sugar is at a 100% concentration in the sugar cube and a 0% concentration in the water. So we would expect that sugar cube to dissolve. And it does. That's diffusion. And that is this diagram is showing you that same thing, but with the addition of a barrier to represent a cell. So let's take it a step further then. Imagine we are a cell, and the cell, or imagine you place a cell rather in a beaker filled with sugar water. And as long as sugar can pass through the cell membrane, which in a real cell it couldn't, but as long as it can, the sugar will go into the cell. The sugar will move down its concentration gradient, and sugar will move into the cell. That is diffusion, but let's look at a different idea. Let's say that now it's real life, and now we place a cell into sugar water. What really happens? Because remember, sugar is a polar molecule, and polar molecules can't move easily into and out of the cell. They actually need, in the body, we need insulin. We need some type of hormone to make things happen, but so that can't happen. If we drop a cell into sugar water, the sugar can't go in. So let's look at a different diagram. Let's look at this diagram here. Same idea. We start here with a higher concentration of sugar on the right and a lower concentration of sugar on the left. Now, if diffusion were at play, these sugar molecules would drift over to the left until both sides of the cell membrane are the same concentration of sugar. But remember, the cell isn't permeable to sugar, so the sugar cannot cross this membrane in this example. So what's going to happen? Nothing? No, because water is permeable. Water can move through this membrane. So where do we have a higher concentration of water? We have a higher concentration of water on the left. So the water is going to move to the right, and that's what's happened in this picture here on the right-hand side. Is the volume of water is actually decreased on the left and increased on the right, and now we have the same concentration of sugar on both sides of the selectively permeable membrane. Now that idea is osmosis, and really all osmosis is diffusion of water or of any solvent, but typically in the body we're talking about water. So if we were to apply the same logic to a cell, if we dropped a cell in sugar water, the sugar can't get in, so the water in the cell would leave and the cell would actually shrivel up. So that is osmosis. So osmosis, diffusion, both of these are the movement of something down its concentration gradient. Osmosis, it's diffusion of water. Diffusion is diffusion of a solute, something dissolved within water. Aquaporins, really quick. So water is polar, so you may have been wondering how is it that water is able to move in and out of the membrane if it's polar, and that is because there are pores for water specifically called aquaporins that allow water in and out of the cell. Osmolarity is just a concentration measure, so you can measure how much of something is dissolved within water by referencing its osmolarity, and that brings us into tonicity. And we've already been talking about this idea of tonicity. So let's say I were, again, sorry to repeat myself, but if I drop a cell into sugar water, the water is going to leave the cell. That's an example of a hypertonic solution. A solution that has a high solute concentration, higher than the cell, is hypertonic. So think of sugar water or salt water. Anything that would cause the cell to shrink because the concentration of water is higher in the cell is going to be a hypertonic solution. A hypotonic solution would be a very pure water. So picture distilled water. If I were to drop a cell of mine into a hypotonic solution, it would actually swell up and potentially lyse or pop. And that's why if any of you guys work in the hospital, if you ever look at just a regular saline IV bag, it's not just water, it's not distilled water, it's a slightly hypotonic solution or possibly isotonic, and it's usually a certain percentage NaCl, or there's more rarely you can have a certain percentage of glucose in there as well. Anyway, the point being a solution can be hypertonic, which means it has a higher concentration of solute than your cell in it, hypotonic, a lower concentration, or isotonic, the exact same. So quizzes, you may see a question like, if a cell is immersed in a hypotonic solution, we would expect its intracellular volume to increase or decrease. So again, if we're putting a cell in a hypotonic solution, that means the water is really highly concentrated outside of the cell and it wants to get in, and it does. So the intracellular volume of that cell will increase. In a hypertonic solution, the intracellular volume would decrease. And then hydrostatic versus osmotic pressure. So I'm actually going to bring this beaker back. So in this first picture on the left, we have osmotic pressure pulling the water from the left to the right. The water wants to go to the right because it wants to go down its concentration gradient. It's just a natural force, and that is osmotic pressure. Now, if we look at this picture on the right, the hydrostatic pressure is trying to push the water back because the water pressure is higher on the right, either due to just pressure or in this case, gravity, because the column is higher on the right, that there is water pressure wanting to push the water to the other side. So hydrostatic pressure and osmotic pressure are often playing a tug of war. This is definitely the case in your capillaries, and we'll be talking about these concepts more as we go. Diffusion, there's different types of diffusion. We've been talking about simple diffusion. So that's just the process of something moving down its concentration gradient. So say we have some lipids in our extracellular fluid. They may just passively and simply diffuse into the cell without the use of channels or pumps or anything like that. Facilitated diffusion is when we start to use channels, or maybe we use little carriers. It almost looks like a pump, but it's not powered by anything. So we're going to save this for a few slides from now. So just sit tight. Okay, we're going to skim this here. Active transport is any transport process that requires ATP. So let me give you, and this, you might want to jot this down. So sodium, let's use the example of sodium. Sodium is 10 times more highly concentrated in your extracellular fluid than in your intracellular fluid. So it wants to get into the cell, right? If it's 10 times more highly concentrated outside, and there are leakage channels for sodium, so it can enter the cell passively, it wants to get in. And it does. It does. It floats in passively. So then how do we maintain a 10x concentration gradient if it's constantly leaking back into the cell? We're going to learn later that we need to maintain that 10x concentration gradient. We need the sodium outside of the cell. And to take it a step further, we need potassium to be 10 times more highly concentrated inside the cell. And they are, fortunately, or else you wouldn't be alive. But they're constantly leaking the other direction. So potassium is constantly leaking out and sodium is constantly leaking in. If we let that happen, eventually there'd be no difference. There'd be no concentration gradients of sodium and potassium, and we would not be alive if that were the case. So we need to use energy in the form of ATP to pump sodium out and to pump potassium in. And that's done through a process called primary active transport. And a great example of primary active transport is the sodium-potassium pump. It pumps sodium out and it pumps potassium in. There are other types of active transport that we won't cover here, but the idea is energy is stored in a concentration gradient and used to move another molecule. I won't ask you guys about that. And then there's vesicular transport, which is the actual cell membrane can wrap itself around something and bring it into or out of the cell. And that's a process called endocytosis if you're bringing something in, exocytosis if you're bringing something out, and then vesicular trafficking is just the idea that we can move these vesicles around within the cell and transport things using these cell membrane spheres that contain different molecules and compounds. And I won't ask you about that either. Okay, here we're getting to something pretty interesting that I'll ask you quite a bit about, and I'm not sure how to play a video within a video, but I would like to show you a video on this. I think what I'll do is I'll post a separate video on aandponline.com called Resting Membrane Potential. It'll be a YouTube link and you can watch that. And I'll do my best to explain it without a visual. Okay, so we're going to talk about this idea of a membrane potential. What in the world is a membrane potential? So if you guys remember from, I don't know, earth science, physics, I don't remember when you would have taken this, but the idea of potential energy and kinetic energy. So if I pick a big rock up over my head, I've now stored potential energy. I've created a gradient. It wants to drop and hit the earth. So I've stored potential energy in it. As soon as I let it go, it falls to the ground. So by picking the rock up and putting it on like the top of a ladder, I've stored potential energy. All I have to do is push that rock off the ladder and it will fall. A membrane potential is the same thing, except instead of a rock, we're talking about charged particles. So say I take a cell membrane, here's one in my picture, and I just put a ton of potassium inside. Say 10 times more potassium inside than outside, which is the case in our cell. I've created a potential, and let's say it's sealed, so there's nowhere for it to go. I've created a potential because that potassium wants to leave. It's highly concentrated inside. It wants to get out. So as soon as I create an opening for it to leave, like a leakage channel, for example, it will. Okay, so you create potential energy by highly concentrated potassium inside the cell. So that's membrane potential. And potassium is just one example. We're going to be looking, though, at a specific example called resting membrane potential. And this is the idea that muscle cells, nerve cells, excitable cells at rest, when they're not being stimulated, display a membrane potential. The inside of the membrane is more negative than the outside. So if we were to open a channel for sodium, let's say, sodium would rush in because sodium is more highly concentrated outside. So we've created an electrical potential by maintaining these steep concentration gradients. So I want to talk about how we got there. How did we create the resting membrane potential? So let's look. So here is a picture of a cell. We have a lot of potassium inside. We have a lot of sodium outside. And we have leakage channels. So we're really only going to be talking about potassium. Sodium's involved as well, but we're really only going to be looking at potassium. That's the key ion in terms of establishing membrane potential. So if these channels are always open, these are leakage channels, where is potassium more highly concentrated? It's more highly concentrated inside of the cell. So where does it want to go? It wants to leave, and it does. Look at the left side of this drawing. The potassium is leaving. Now, potassium is a positively charged ion. So as it leaves, it's pulling with it negatively charged proteins and anions that are just sort of following it because opposites attract, right? So they're sort of following it out of the cell, but then they get stuck. They can't leave. So not only are we losing all these positively charged particles, we're collecting all these big negatively charged particles inside this membrane right here. So we're losing positive, we're gaining negative, so we're starting to develop a negative charge. Let's say this is a neuron around negative 70 millivolts or so, right? So say we start at 0 millivolts difference between the inside and outside. But as the potassium leaves and as the anions accumulate on the inside of the cell membrane, we get to negative 10, negative 20, 30, 40, 50, 60, 70. So now we're at negative 70 millivolts. Well, it's getting really negative inside the cell. And potassium is a positively charged particle, so it's going to start to get pulled back in. So now we have two forces at work. We have a concentration gradient pushing potassium out, and we have an electrical gradient pulling it back in. At some point, those two forces will be in equilibrium. And whatever that value is, so let's say the electrical force pulling it in is equal to the concentration force pushing it out at negative 70 millivolts. Let's say it's negative 70. that is the resting membrane potential. The resting membrane potential is -70. At rest, what is the membrane potential between the inside of the cell membrane and the outside? That's the resting membrane potential. And that number is achieved by what type of electrical force do we need to keep this reaction in equilibrium? So to be equal to the potassium force, the concentration force forcing potassium out. So it could be anywhere from -40 to -90. It just depends on the cell. But -70 is used a lot when we're talking about neurons. So anyway, that is resting membrane potential. It is achieved by the steep concentration gradient pushing potassium out, creating a negative force here inside the cell. And then the reason why it just doesn't keep going up to -80, -90, -100, -110 is because at some point the potassium gets sucked back in by an electrical gradient. So we have a chemical gradient, a concentration gradient pushing it out, and an electrical gradient pulling it back in. None of that could work if it wasn't for the fact that we are constantly forcibly pumping potassium in and pumping sodium out. Remember, potassium is 10 times more highly concentrated inside than outside. If it wasn't for that fact, it would never get pushed out to begin with to create this negative force. So test questions on that. I may ask you, what ion is most important for establishing resting membrane potential? It's potassium. Where is potassium more highly concentrated? It's more highly concentrated inside. I may ask you, and I haven't even told you yet, so I'll tell you now. Is membrane potential, does it mean that the inside, the whole inside, the sum of all the electrically charged particles inside the cell is 70 millivolts more negative than outside? Or does this charge separation exist only at the cell membrane? And the answer is it only exists at the cell membrane. If we were to add up the particles of the intracellular and extracellular fluid electrically, we would find they're equal. This charge gradient exists only at the membrane. And it may kind of seem like who cares right now, but your neurons wouldn't work and your muscles wouldn't work if it wasn't for this fact. We need the inside of the cell to be slightly more negative at the membrane at rest than the outside in order for your brain to work, your heart to work, your muscles to work. Okay, we're going to go ahead and skip this slide. We're going to come back to this so much when we get to endocrine, so we'll just let that go as well. So let's look at the cytoplasm. We have cytosol. This is the fluid that everything is floating in. Organelles are the metabolic machinery of the cell. Inclusions are just some other thing that's included within the cell, and it is going to be based on cell type. So let's say we're talking about muscle cells. We're going to have lots of myoglobin stored within the cell. That's an inclusion. If we're talking about a fat cell, we're going to have a big droplet of triglyceride stored within the cell. That's another inclusion. Let's go ahead and just look at the organelles. So the first organelle we're going to talk about is the mitochondrion. This is kind of the star of biology classes everywhere, so you've probably talked a lot about this in the past. We're going to keep it very basic. It is a membranous organelle. It actually has a double membrane. It has two membranes. So the job of this organelle is to take pyruvate, which is a byproduct of glycolysis or anaerobic respiration, to take pyruvate and turn it into ATP, water, and carbon dioxide. So you take oxygen and pyruvate and turn it into ATP, water, and carbon dioxide. This is where aerobic respiration occurs. It's where the bulk of our ATP or cellular fuel is produced. I forgot where I heard this, but I think it was on the radio or a podcast that I was listening to. And the question was, where do you lose weight? So I'll pose that question to you guys. If, say, you lose 10 pounds, where did it go? Where did you lose it? And I always ask that in class, and I get a lot of funny things. People usually start with, well, it's converted to energy. So I lost 10 pounds. That 10 pounds was converted to energy. It's not technically true, though. Only a nuclear reactor can convert mass into energy. our bodies aren't nuclear reactors. They're pretty amazing, but they can't convert mass into energy. That's impossible. So then from there, people have a funny thing, like you lose it in your poop. No, you don't. You actually lose your weight through your nose or your mouth, if you're a mouth breather, I guess. You lose your weight through your nose. All 10 pounds of weight, or most at least of the 10 pounds that you lost through your nose, which people are like, what? And so was I. So imagine this. You breathe in O2, 2 molecules of oxygen. You breathe out CO2. Which one weighs more? CO2, obviously, right? Because you breathe in O2, you breathe out CO2. There's an extra carbon atom in each molecule of CO2 that you breathe out. So every time you take a breath, you get lighter. Not much lighter. So when you lose 10 pounds, that weight is actually lost through the loss of carbon, which is a byproduct of aerobic respiration. And what makes you perform more aerobic respiration? Well, burning more calories, or exercising, or maybe you just eat less, so you're bringing in less weight than you're breathing out in the form of CO2. So I heard you all to start breathing faster, trying to lose weight. I'm going to start hyperventilating. Anyway, so the mitochondria produces ATP. It's the site of aerobic respiration. These are interesting little guys. I don't want to get down like any type of philosophical discussion or anything about mitochondria, but they are odd. They're odd. And what I mean by that is they have their own set of DNA, and that DNA is actually very similar to a specific type of bacteria. They kind of look like little bacteria. They move around on their own. They're responsible for their own replication, and you inherit the DNA from your mitochondria, not from both of your parents, but just from your mother. You inherit the mitochondria from the egg cell, and then it divides as your cell divides. It's fascinating. If you want to get into an interesting line of research, figure out where mitochondria come from, or not figure out where they come from, but just figure them out. They're just an interesting little organelle. For the test, what I want you to know is that they produce ATP. They're the site of aerobic respiration specifically, and that they're a membranous organelle. The next structure here is the ribosome. The ribosome is their subunits are produced in the nucleolus, and their job is to make proteins. So they transcribe messenger RNA and turn that into chains of amino acids that then are modified and fold into more complex proteins. So ribosomes are non-membranous organelles that make protein. Next, we have the endoplasmic reticulum. You have 2 versions, a smooth and a rough ER. A smooth endoplasmic reticulum, it metabolizes lipids, it stores calcium, it's responsible, let's just say, for lipid synthesis and calcium management. The rough endoplasmic reticulum, it's rough because it has ribosomes attached to it. The rough endoplasmic reticulum packages and modifies proteins produced by ribosomes. The next structure is the Golgi Apparatus. The Golgi Apparatus further packages proteins that it receives from the endoplasmic reticulum. So a vesicle filled with protein will So a vesicle filled with protein will arrive from the endoplasmic reticulum at the cis face. That's the receiving side of the Golgi apparatus. It will be modified, maybe sugar groups will be added, things will be packaged differently. And then it leaves the Golgi apparatus for export from the cell on the trans face. That's the shipping side of the Golgi apparatus. This next 2 structures, they look somewhat similar. They're called peroxisomes and lysosomes. Peroxisomes, through the use of oxidases and catalases, use molecular oxygen to detoxify harmful substances like alcohol and formaldehyde. And then, like I said, they also neutralize free radicals, which are highly reactive chemicals with unpaired electrons that can do damage to DNA. Lysosomes they look similar. They're vesicles, but they have proton pumps in them. And if you remember from chemistry, how do you measure how acidic something is? Well, you put it in water, and then you measure how much hydronium, which is H3O, is produced from the addition of that substance. So you say you put hydrochloric acid in water, and you say, oh wow, it makes a lot of hydronium because the hydrochloric acid donates hydrogen atoms. So hydrogen atoms are then acidic, if you think about it. A hydrogen atom specifically, if you remove an electron, is just a proton. So if you want to make acid, one good way would be to add protons to something. And that's what the lysosome does. It has a ton of proton pumps, and it pumps protons into the inner lumen of this lysosome, and it makes a very high acidic compound. And there's different hydrolytic enzymes, and it's just bad stuff. You don't want to end up inside a lysosome. So a lysosome is basically the stomach of the cell. So let's say I'm a white blood cell, I'm a neutrophil, and I phagocytize some bacteria. So I bring this bacteria inside the cell. I have this vesicle filled with bacteria. I'm going to send some lysosomes over there to merge with that vesicle and release acid into and onto that bacteria to digest it. They also digest dead organelles and just anything that needs to be digested. So peroxisomes and lysosomes are both membranous organelles, and as are, Golgi Apparatus and Endoplasmic Reticulum. These are all membranous organelles. Now we have some non-membranous organelles. These are the different rods that make up our cell skeleton. We have three types. We have a microfilament. It's the smallest in diameter. You're going to find a lot of these just deep to the cell membrane. It's called the terminal web. It's like a spider web of these little filaments that give the cell its shape. And then motor proteins can tug on these filaments to make the cell change shape or divide or move around. Intermediate filaments are slightly larger. They have a high tensile strength. You can kind of think of them as rope. So the intermediate filaments are going to be anchored to the desmosomes, and they're going to hold that cell together. They have high tensile strength. And then lastly, the largest of the structures is called a microtubule. Microtubules do a lot of different things. They're kind of like railroad tracks for your different organelles or vesicles. Things can move around using your microtubules. They're like the roads of the cell. They can also be used to force the cell apart during mitosis, and we'll be taking a deeper look at that. All of those microtubules radiate somewhere from the center of the cell called a centrosome. And a centrosome is a non-membranous organelle that's composed of two centrioles. Centrioles are just these odd-looking barrel-shaped tubule structures. There's two of them, and they're oriented at a right angle to one another, and they're surrounded by microtubule subunits. And the microtubules grow out of the centrosome.